If it's
That's
not
it.
going to work,
Look,
it's not
there you go. It's that.
It's doing it?
Yeah.
Is that recording now?
It's got a red button. Look.
Okay.
yeah.
Well done, girl. Okay. Okay, brilliant.
Shall I move his chair down?
Might not be the best angle now. I remember doing that before.
He's over there, not in the picture.
Oh shoot, this isn't going to work here then.
No, that's all right.
Just can't see him.
Welcome everybody today. We've got some new faces. Lovely to see everyone. Some faces not seen for a while. So thanks everyone for coming.
Just move that chair.
Just let you know the fire exit is actually behind here.
Perfect. Put it in front of the thing and then nobody can help.
There's the car just there where you need. Next to the toilets, you go through the kitchen door and you turn left and they're just there. No, that's right, sorry. Through the kitchen door, turn right and the toilets are just there. Help yourself to food and drinks. I need to thank Mike who's funded today. Sohel who's organised and done all the liaison. And of course thank you Dr. Shephard for coming today.
Well, thank you very much.
We are recording the talk, so we've got a video recorder and a voice recorder as backup, which I'll start now. Okay. So we're voice recording it as well, and the aim is to send it out to by email and on the website, so for all the people who can't get here, they'll be able to have access to it. And let's see, what else is there? So if everyone checks that they, if they haven't signed in, just sign in at some point. And also, let's see, so the running will be, Dr. Shephard's going to do a presentation for about 40 minutes, and then we're going to have a 10-minute break, and then do questions and answers. Mike, I'm going to hand over to, so he's going to chair the meeting because he's a lot more assertive than me. So I'll pass it over to him, and then he can tell you just how it's going to run, and then ask you.
Yeah, great.
So thank you everyone, thanks for coming.
Well done, Maxine. This is all Maxine's hard work. We'll appreciate that, Maxine. Thank you very much indeed for everything you've done. Charles is going to speak for about 40 minutes, 10-minute break, then some questions. I will say this again, I want questions, I don't want statements, we don't want stories, we want some questions because we need answers. And so let's deal with that when it comes. Dr. Shephard needs to be away by 4:15, so that's the timing. And what I don't want to hear at the end of the meeting is someone say to me, well, I really wanted to ask this. You know, this is your opportunity now to ask some questions when Dr. Shephard's finished. Thank you very much for coming. It's all yours.
Can I add something?
What?
We have got the room booked till 5, so although it will be finishing at 4 as the talk, there's an extra hour afterwards if people want to have a chat with each other and that sort of stuff. And does anybody mind if I don't share photos? I wanted to share photos on the website and newsletter. So if you're not okay with that, please let me know. Come and see me afterwards. Okay, thank you.
Right, shall I kick off then? Yeah, great. Right. Well, thanks everyone who's organised this. I ought to mention Sohel, who's brought me here from Watford Station, because I've been doing all the email correspondence with him, and it sounds like you've got a very efficient setup here, so that's really
Can I ask one favour? Can you just move a couple of feet? Because at the moment I'm not getting anything on the
You just
video.
turn it.
Oh, right.
No, because there's someone in the way. Was it really going to be
You're in charge of the video?
only temporary? That's perfect.
Right.
That's perfect.
I better just warn you, I'm one of these people who wanders.
Oh, if you're wandering,
around.
that's fine.
So I'm going to be wandering from there. And
No,
I'm sort of wandering
that's
to their
fine.
point.
We're good now.
Right, okay. Right, okay. Right, so what am I going to do today? As has already been said, I'm going to speak for about 40 minutes. I decided what I would do, rather than give you a talk, which some of you may have heard from other people, you're probably fairly up to speed with it in any case. I'm not going to sort of give you a talk on how to diagnose ME and how to manage it. What I'm going to do is give you a talk on basically everything that's going on at the moment from the point of view of management, research, politics and everything else. We've got an hour for questions and answers afterwards. I've brought quite a lot of literature with me which covers a whole range of management things, so I'm sure we can get into management, specific sort of management issues like pain, sleep, whatever, during the questions and answers. So I'm going to try and concentrate on a pretty rapid tour of everything that's going on at the moment. If you've got questions and answers, they can be related to what I've been talking about or anything else that's going on that I haven't even mentioned. If there's anything that I bring up during the talk that just requires some very quick, brief clarification instead of leaving it to questions and answers, either just shout your hand up or say, what's that? Or you didn't mention that bullet point or whatever. But otherwise, try and leave it to Q's and A's. For people who are not familiar with how I got involved in this condition, I've had this wretched illness for almost 40 years. And like many people with it, I was an extremely fit young adult. I was working in hospital medicine. I was dealing with a person who had a very nasty dose of shingles. That's the chickenpox virus. I called a very nasty dose of chickenpox. I had what's called an encephalitic component to this. I was really quite ill with it. That means I had virus on the brain. It affected my cerebellum, which is the part of the brain that is involved with balance, and at one point I had to go to the National Hospital in Queen's Square, the big neurological hospital in London, to try and get things sorted out. So I went through the process that I'm sure many people here have been through of trying to get a diagnosis, trying to get some help from doctors, and I went back to work, I went off sick, went back to work, went off sick, and because my own illness was so badly managed at the start, over a period of 2 years, it was almost 2 years before I actually went to see Melvin Ramsey, who was the doctor who first described this illness at Royal Free Hospital back in 1955, got a diagnosis, and was told how to manage it as best I could at the time. That early initial stage of not having good management, not having a diagnosis, and doing lots of things that weren't really right, I think had a very negative impact on my own health, and I know the same applies, I'm sure, to many people here. So I've been involved with this illness for, as I say, almost 40 years. I've been a medical advisor in an honorary voluntary capacity to the M.E. Association for probably about 25 years now, probably a little bit longer. I have many other hats on which I won't go into, but the one there in brackets, because we no longer have the MRC expert group, but I was part of the MRC, the Medical Research Council's expert group that was set up to produce a report and produce some ring-fence funding for biomedical research into this illness, because the Medical Research Council, as I'm sure anyone who knows, who follows research into this illness, has not had a very good record when it comes to the sort of research that it's funding. But there has been a significant change in attitude at the MRC since that group was formed, and it is, as I'll tell you in a few minutes, being asked to fund a very big major research strategy proposal that we have put to it. I'm on the board of what's called the CMRC, which is the CFS M.E. Research Collaborative, which followed on from the demise of the MRC expert group. And this is a big sort of umbrella group of really everyone in the UK who is doing research or is interested in research into this illness, the various charities that fund research, and the various funding bodies like the MRC, the National Institute for Health Research, and also drug companies that we're trying to get involved in getting interested in this group. So that's an important research initiative. I also do quite a lot of work on benefits. I belong to a group called the Fluctuating Conditions Group, which was set up by the Department of Work and Pensions in an effort to try and make the criteria for awarding benefits, particularly ESA and PIP, more fair for people who have what are called fluctuating medical conditions. So that's not only people with ME and CFS, but people with multiple sclerosis, hepatitis C, AIDS, HIV, Parkinson's disease, a whole range of conditions that you can't just take a snapshot of someone and decide whether they're fit or not for benefits. We've made numerous recommendations as to how ESA and PIP could be improved. Some of them have been taken board, many have not, and as many people around here will know, trying to get ESA and PIP can be a very frustrating business. More recently, my most recent extra hat, is to belong to the NICE Guideline Committee, which I'll come back to in a minute. And as I'm sure everyone is aware, this wretched guideline on MECFS, which has been sitting there for 2007 and which recommends cognitive behaviour therapy and graded exercise therapy as an automatic treatment for everyone with mild or moderate MECFS, is at long last being revised. It's not only being revised, in actual fact, it has been decided, and when it was decided that it was going to be revised, that it would actually be rewritten. So we're in the process of rewriting the NICE guideline on MECFS, and that will hopefully appear in October next year, 2020. Right, so the first thing I'm going to cover, and as I say, this is a pretty quick tour around a whole range of topics, so it's buttered fairly thinly. But as I say, we can go back to questions and answers if people want to raise more detailed points than anything that I'm talking about. So, from the research point of view, I'm just going to talk a little bit about what's called the MECFS Biobank, and diagnostic biomarkers, drug treatments which are aimed at the underlying disease process, and any other bits and pieces that's going on in the way of research at the moment. That's a picture of the containers, the very low temperature containers in the lab at the Royal Free Hospital in London, where we have our MECFS Biobank. Who's heard of the MECFS Biobank, or knows about it, or knows something about it? Good, good. And I know Sir Hale has actually donated a blood sample.
Oh, wow.
So, very simply, this is a very important piece of new research infrastructure, which the ME Association funds all the basic running costs of the ME Biobank. I steer the ME Biobank steering group, and it is a place, as I say, it is part of the main University College London Biobank, which has a whole series of what we call sub-biobanks for different diseases, and all these samples are kept in this biobank in these very cold storage containers like that I've shown you, in a special place, special building at the Royal Free Hospital up in Hampstead. And we have been developing the MECFS Biobank over the past probably 3 or 4 years now. So we collect blood samples from people with very carefully assessed and defined MECFS, so they have to meet Canadian criteria, Fukuda criteria, and we take a much more detailed history than that, investigations, examination. So we have a vast amount of clinical information on everyone who donates blood samples. So everyone has a very thorough clinical assessment. The samples are then taken. They then have to be couriered very quickly because the sort of information that we're looking for in these samples can go off very quickly. It's not fixed and then frozen. So the samples are couriered to the Royal Free Hospital, the UCL Biobank there, where they are processed and then stored and then released to researchers who want laboratory-based samples, blood samples, and clinical data attached. And it's a far more effective and quick and cheaper way of doing research if that's what you need. You just want blood samples, clinical data. You don't need to actually see the patients. So for researchers who want to do laboratory-based studies, this is a very useful research source. So we now have samples, blood samples, moving to Hales, from over 500 people with MECFS. We have a severe cohort, so we have a research nurse who goes and sees people with severe MECFS at home and takes blood samples from them at home. A group of people with severe bedbound ME in this sample. We have a group of people who act as a control group who have multiple sclerosis, because MS has quite a lot of overlapping symptoms with MECFS, including fatigue, and healthy controls. And that means we now have what are called over 30,000 aliquots, that's subsamples, which means different parts of the blood sample makeup. So people can have different types of cells, different types of components of the actual blood sample. So these samples are now being released to researchers here and abroad. And if you go to the M.E. Biobank website, you will see where we're sending samples to. I mean, at the moment they're going to Israel, South America, North America, the UK, Europe, for researchers who want to do this type of research. We have, because this is recognised as probably the leading resource of this type in the world now, we received over $1 million from the National Institute of Health in America, which is the prestigious American research funding organisation, to carry out a long 3-year study into virology, that's viruses and immune system function in ME/CFS, and one of those papers has just been published. Another paper that's come out of the Biobank recently as well, looking at all the different tests which we can do internally there, has shown that people with more severe ME appear to have, certainly on the samples that we have, a lowered level of an interesting enzyme called creatine kinase. And this is an enzyme, it's a chemical substance which is released from muscle when it's damaged or not functioning properly. So that's quite an interesting observation. We continually need, in sort of stop-start basis, blood samples from volunteers, and it's nice to know that the ME community is so willing to help with that. We don't require any more blood samples at the moment, but when we do require some, we put a message on the MEA website, in the magazine, on our Facebook page, and on the ME Biobank website. So one of the things that obviously people want to do with blood samples is to look for what are called biomarkers. So we're looking for abnormalities, consistent abnormalities in the blood, which are going to help us with making a diagnosis of ME, because I'm sure, as everyone knows, one of the major problems of getting a diagnosis or not getting a diagnosis of this illness is the fact that we don't have a blood test to diagnose ME/CFS. So there's a real big search going on both here and in the USA to try and find what we call a diagnostic biomarker. For those who follow research, that's Professor Ron Davis, who I'm sure some people here have heard of over at Stanford. He has recently published, well, not that recently, it's quite a few months ago now, but he has published some results from one of his biomarker studies using immune system analysis, and claiming that he has actually found something which may be a useful diagnostic biomarker. But the problem with biomarkers is, although they can help us with diagnosis, perhaps subgrouping under the ME/CFS umbrella, and even understanding the underlying cause of a disease, we do need to have robust information. So we need information from really large numbers of people. The results have to be consistent, and they have to be present in ME/CFS, but not present in other diseases which cause fatigue. So we want to make sure that that abnormality is something that we find in people with ME, but it's not something we find in people with MS and depression and everything else. So at the moment, there is a lot of research going on in that direction. We're also using some new investigative tools called metabolomics and proteomics. Metabolomics is a way of looking at what we call metabolites. They're chemical byproducts of things that go on in your cell. So using these blood samples, we can look for abnormalities in what we call metabolomics, and we can also look for abnormalities in proteins in the blood, and that's proteomics. So these are two important, relatively new investigative techniques that are being used in the analysis of these blood samples from the biobank. So as I say, how far are we on with getting a diagnostic biomarker for ME/CFS? There is the NIH study at the biobank. We didn't, like the Australians have done, found an abnormality in natural killer cells in these patients, but we did find an abnormality in something called MATE cells, which are mucosal associated invariant T cells, very small print stuff. But it seems to be that there is a specific abnormality in a part of the immune system in these patients. Second one, again, as I mentioned, it's interesting to find low creatine kinase, this muscle enzyme in people who have severe ME/CFS. Ron Davis at Stanford done 2 studies which have been well publicised, but really are preliminary information at the moment. The first one was on changes in the shape of red blood cells, and then the second one was what we call this nanoelectric study, which came out, I'm trying to think when it came out, earlier this year, on what in their terminology was an immune system stressor reaction. One other test that I will mention is the Acumen test. Don't know, anyone familiar with the Acumen test for mitochondrial function, dysfunction? Yes. This is a commercial test which claims to pick up abnormalities in mitochondrial, that's muscle energy, function. And this is being produced and sold commercially. We have had concerns about it, as have many of my colleagues in the medical profession. It's a commercial test which is not used by doctors who are interested in muscle and mitochondrial disease. And so we asked our colleagues that we fund at the University of Oxford, that's Dr. Carl Morton and Professor Julia Newton up in Newcastle, if they would carry out a big study to try and validate the Acumen test. And they failed to do so, and those results have now been published. So I think one has to conclude that at the moment, on present evidence, the Acumen test is not an accurate test for detecting abnormalities in mitochondrial function, and it is not something that we would recommend that you go and spend money on. If you want to find out any more information about all these different studies that are going on looking for these biomarkers, all these studies are reviewed. We review all the key research that is coming out on the MEA website, and it goes on our news carousel. You just go to the MEA website, you can go into the news archive, and you'll find the papers and a lay explanation as to what they mean. So in conclusion about this search for a diagnostic biomarker, in order to draw firm conclusions about a diagnostic test, we need to do this in large numbers of patients. We need to compare these results with other diseases that cause fatigue, and then most important of all, when we've got some sort of abnormality like Ron Davis has found or someone else has found, it needs to be replicated by other independent research groups. So none of these tests are available on the NHS at the moment, but of course you can get your creatine kinase done, of course, on the NHS. It's something that should be done in the diagnostic process. But I think we are some way off from actually getting a diagnostic marker blood test for this illness. Now, the other thing in the way of research that's going on is obviously this search, because we want to find a treatment for the underlying disease process in this condition. We have treatments which can help with symptoms, particularly pain and sleep, but we don't have a drug which can help or treat or cure the underlying disease process, partly because we're not quite sure what the underlying disease process is. We know this illness starts with an infection, but it doesn't appear to be due to a persisting viral infection. We know that that infection triggers an abnormal immune response, and I've referred to some of the abnormalities in the immune system that we know about. And it seems at the moment that that abnormal response that's triggered by the infection, the triggering infection, is what we call an overactive immune response. Your immune system goes into overdrive, as it would do when you're dealing with a dose of flu, and then doesn't settle down again. So one of the approaches to possible treatment of this disease is to use the sort of drugs that you use in rheumatoid arthritis and other immune system diseases where you've got overactivity, that type of immune system dysfunction, to see if you can dampen it down. So that's the Norwegian doctors, Fluge and Meller, who you're probably familiar about if you followed the story of rituximab. And they, we're going back 3 or 4 years, found that some of their patients with Hodgkin's disease, who were given a very powerful drug called rituximab, which dampens down a specific part of the immune system called the B cells, and dampens down the activity of what we call autoimmunity. That's the body's immune system turning on itself and producing antibodies against its own tissues. They found that these patients who also have ME improved with rituximab. So they did a clinical trial on rituximab. In fact, they did 2 clinical trials to start with on rituximab, and they were produced reasonably encouraging results. But sadly, people who followed this story will know, and the results, the full results from the phase 3 clinical trial were published this year. When they did a big multi-centre clinical trial of rituximab, it failed to confirm those results. And sadly, I think at the moment that is the end of the end of the road for rituximab. But there's certainly other what we call disease-modifying drugs around which are worth following up. And certainly because we know that these immune system abnormalities that are there are other types of immune therapy which are worth trying. It is possible, having said that most of the evidence is pointing against persisting viral infection in this illness. What we do know is that some people seem to, possibly as a result of the immune system disturbance, stir up what we call latent viral infections in the body. So there are infections like Epstein-Barr virus, which is the glandular fever virus, and certain types of herpes viruses, which once you catch at some point in your life, just sort of hang around in a dormant state. And there is some evidence that as part of the disease process, you may be stirring up and reactivating these particular infections. So there may be a role in certainly a subgroup of people to use antiviral drugs, providing that you've got good clinical evidence. And there's a research group also at Stanford University in America, headed by Professor Jose Montoya, who's not there at the moment, for reasons I won't go into, but they have used these drugs quite successfully in a small subgroup of people who appear to have reactivation of these herpes viruses. So at the moment we can't rule out the use of antiviral drugs. There may be some forms of immunotherapy which are worth trying, but the bottom line is here we do not have a drug which we can use to treat the underlying disease process in ME/CFS. Having said that, and as I said, I'm not going to talk in any detail in this talk on management. The most crucial aspects of management of this illness at the moment, in the absence of having a drug that can treat the underlying disease, the most crucial parts of that management are activity management, which in our view means pacing as opposed to graded exercise, and dealing with specific symptoms, both using drug treatments where necessary and self-help where necessary as well. We have at the ME Association, I've brought some of these leaflets along today. We've got specific leaflets covering pain, irritable bowel syndrome, postural orthostatic tachycardia syndrome, sleep disturbance, all the different symptoms that are associated with ME/CFS. You can download all these leaflets from the MEA website if you want. And in the case of pain, we also have separate leaflets covering the various drugs which can be useful in the treatment of pain, particularly things like amitriptyline, which some people may well be on here, low doses of amitriptyline, gabapentin, which is a drug which is normally used to treat epilepsy, pregabalin, Lyrica, which is another drug which is used sometimes in more severe pain relief. So we've got a whole range of literature on the management of individual symptoms for those who want to follow that up. As I say, I've brought some leaflets here. They're all on the table there. They're all free, apart from the purple books, which I do have to make a charge for. They're quite costly to produce. The other developments I quickly cover in the way of research. As I say, we have a research collaborative which I sit on the board of. We've got a 2-day conference in Bristol in March next year. Details are on the MEA website. The public are more than welcome to come along to our conference. It's not that expensive. You're made very welcome. So if you want to come along to a research conference, check that out. We have, as I briefly mentioned earlier, been in the process this year, and there are some details on the website. There's a big question and answer on what this research strategy proposal consists of, but we have just submitted, and we've got a big meeting on November the 25th to take this forward, a large, this is a multi-million-pound research strategy proposal which is going to the Medical Research Council and the National Institute of Health Research. And there are two components to this. We want to do a 20,000 patient, what's called a GWAS study. And this is going to involve collecting saliva samples from about 20,000 people with ME/CFS and looking, these will then go to the National Bioresource Centre in Milton Keynes for processing, and then the DNA will go up to Edinburgh where Professor Chris Ponting will look for genetic abnormalities, genetic clues as to the cause of this illness. And the second part of this study will involve extending the number of people, the samples we have at the ME Biobank, from around about 500 up to about 1,000, so that we can carry out more detailed work looking at the new technologies I described earlier, the metabolomics, the proteomics, the small print stuff of what's going on in the blood. One final piece, and it's a sad item of research, but I will just mention it because it's one of my particular bits of research that I'm involved with. We also fund and facilitate post-mortem research looking at tissue from people, sadly, who have died of ME/CFS. And this is carried out at Addenbrooke's Hospital in Cambridge, that's the big teaching hospital at Cambridge University, where Dr. Dominic O'Donovan, who's a neuropathologist, a pathologist who just looks at nervous tissue, will look at brain and other tissue from people who have died. Sadly, in most circumstances, this is people who've died acutely from suicide or other acute causes. So he will look at this tissue if it gets to him in time. And one of the interesting things that we have found looking at this tissue is that there's a part of the nervous system which lies just outside the spinal cord, and these are little, they're tiny in actual size, but in number they contain large numbers of neurons, nerve cells, and they're called the dorsal root ganglia, and they're sort of like the Clapham junction of the nervous system, where lots and lots of incoming nervous activity is all gathered together and coordinated. And there appears to be inflammation within these dorsal root ganglia, and that's called dorsal root ganglionitis. And we've published those findings, and if you want to follow that up, it's in the purple book there. As I say, we have published those findings in a research journal.
Excuse me,
Yes.
sorry,
Yeah.
before I forget, can I just ask you, how are the 20,000 going to be recruited, new volunteers? And the other one is, can you leave your body for post-mortem tissue?
Yeah, two good questions. So, if this study gets funded, and we will put in the actual funding application in January,
Right.
and then hopefully we will get notified towards the beginning of next year. So if it is funded, we will there will then be a very much a sort of social media recruitment campaign. Because the numbers are huge, we won't have the facilities to actually send a physician out to examine everyone to make sure they've got ME/CFS. So they will be diagnosed, because we do want to make sure that people have got ME/CFS as best we can. They will be diagnosed using a diagnostic tool which has been developed at the ME/CFS Biobank for preliminary diagnosis of patients who come to give blood samples. So they will be asked to give a comprehensive clinical history, a sort of tick box thing. So you'll have to have post-exertional malaise, etc., etc. You'll have to have symptoms, enough symptoms to fulfil on paper a diagnosis using Canadian Fukuda criteria. So we're doing as best we can with a huge number of people to make sure that the diagnosis is correct. But it will be open to everyone. It will be widely publicised. It will be very simple to do. It's the saliva sample is basically actually spitting into a sample container and sending it off to the National Bioresource Centre. So there's
So if you've got a medical diagnosis of ME?
Yes, yes. I think in addition to having, I think we've got to work this it's not absolutely finalised at the moment. So the first stage may be, have you got a firm diagnosis from your doctor, and then we will ask you to tick box off the diagnostic
So
criteria.
will that be in the magazine? Sorry.
It'll be in the magazine, it'll be in the website, it'll be all over the place. It'll be very simple to do because we've got to reach out to a very large cohort of the patient community, and that we are actually discussing whether we might even take samples from abroad for that part of the study. The second bit, how do I donate to post-mortem research? A very important, very good question, which I should have mentioned. If you go to our website, you can download what's called a statement of intent, and you fill that in, that goes with your will and whatever, and that makes it clear, because one of the hurdles of doing post-mortem research is that sadly we hear that someone's died, and then the relatives say, well, I think they wanted to give their body, if they did die, to research, or it says, has anyone written it down anywhere? And if it's not written down clearly, it doesn't prevent it, but it makes it a lot more difficult. And the problem with post-mortem research, it's rather like the blood samples, we really need to, from the point of death, to remove the samples and get the crucial samples to Cambridge. That really needs to be done within 48 to 72 hours of death. So we want to know as soon as possible and get things moving as soon as possible. And it does help, obviously, if we've got a statement of intent. So that can be added to it. So that's that you can download from the MEA website. So that's
can I ask,
yeah,
is that the
yeah.
same as Mega? Because I read about a study called
Mega,
Mega.
well, Mega, this whole thing started, Mega, as you know, failed. And Mega was, part of the Mega was a GWAS study, but this is rather different to Mega. The people involved, without going into personalities, are different to people who were involved in Mega. And if you, have you had a look at the website Q&A on this?
I just saw the story about it, but I
Yeah, well,
confused if it
we've
was
done
Mega
a big
or-
Q&A, we've got a patient involvement group. We're really trying to get the patient community involved in this to make sure that the patient community is happy with what's going on, that is fully informed. So there is a patient